Let me add the, so here this is the another H here, so same way we have our phosphates here, negative. Okay, so same way. Okay, let me draw this properly. CH2. Okay, so now let's call this is our 5 prime end. This is our 5 prime end here, and this is our 3 prime end here. So can you see? So now this is our 5 prime here, because this one is on the 5th carbon here. The phosphate group is on the 5th carbon, while the phosphate group is on the 3rd carbon. So now let's assume it's moving in this direction. Now are you getting it? Now I said earlier that thymine must be paired with adenine. So this adenine here, thymine must be here. Are you getting it? Now for thymine to pair with it, what really has to happen is that they have to run anti-parallel to themselves. Are you getting it? So this is a strand now. Now the other strand that will join to it has to run anti-parallel. Anti-parallel in what sense? In the sense that it must start from the 3 prime end and end with the 5 prime end, or like the other one that starts with the 5 prime end and then with 3 prime end. So now let's go. Okay, let's assume this is our thymine. Okay, so I wouldn't try drawing much of structure. Okay, this is 0. So this is our carbon. This is our carbon 1. Okay, this thymine. Are you getting it? So the same way. So it goes like that. Carbon 1, carbon 2, carbon 3. So this is our phosphate group. So this is our phosphate group here. Let me represent with P. So the same way like that. Also on our, after the phosphate group, what else do we have? Okay, this is our nitrogen base. This is our phosphate group. Okay, so it goes like that also. Are you getting it? So it goes like that also. Another phosphate group is joined just the same way here. So it runs anti-parallel. So this is the 3 prime end. This is the 3 prime end. 1, 2, 3. The phosphate is joined to the 3rd carbon here. So finally, just as we have here in the previous one. Are you getting it? Finally, 1, 2, 3, 4. Finally, our phosphate will be here. Are you getting it? So can you see, just like the other way around of it? Now let me quickly show you something. So this is what we mean by anti-parallel. So thymine will be here. Then what else do we have? Okay, cytosine and guanine will be here. So the same way like that. Now look at something. Let me quickly show you. This is the from here to here. Here we have this is the phosphodiester linkage. So phosphodiester linkage is joining the sugar and the phosphate group together. That's the phosphodiester linkage. So from here to here, now look at it. The O here, the here. So it's joining the sugar and the phosphate group together. Phosphodiester linkage. So also we have which one again? Okay, yes, the bond between. Don't forget that the base is always on the carbon 1. The bond between the base and the sugar. That's glycosidic linkage. So then the bond between the bases, like the bases thymine and adenine, that's hydrogen bond. Are you getting? So it goes like that. So let's finally, having done that, let's talk about the purine and pyrimidine. Now purine and pyrimidine, they are organic bases in DNA and RNA in nucleic acid. For example, now in DNA, DNA could either contain purine or pyrimidine. Are you getting it? So now purine is made up of adenine. Purine is made up of adenine and guanine. So why pyrimidine is made up of cytosine and thymine. Are you getting it? So that's in DNA. In RNA, purine is made up of adenine and guanine, same way in DNA. But don't forget, in RNA we don't have thymine. So as a result of that, it will be made up of uracil and cytosine. That's pyrimidine in RNA. So that's fine. And now, before I end this video, let me show you a simplified